glycoconjugates. As I mentioned earlier, when besides carbohydrate, other things are attached, either protein or lipid or any other group is attached with the carbohydrate, then it is called glycoconjugates. Let's describe it. Proteoglycans.
Yes.
Glycoprotein.
Yes, sir. Yes.
Let me describe this thing. Glycoproteins. Oligosaccharides joined covalently to a protein. Particularly carbohydrate is attached with the oligosaccharide portions are very heterogeneous. Actually, when it is, as I mentioned, 2 to 20 monosaccharides, if they are together bound with glycosidic bond, then they are oligosaccharides. And there can be some modification, either N-acetyl or sulfatation. So that heterogeneous is due to that. Found on the outer face of the plasma membrane, extracellular matrix and blood. Inside the cells, this I explained it also. Inside the Golgi. Actually, we were discussing here only. Inside the cells, it is found in the Golgi complex, secretory granules and lysosomes. Some cytosolic and nuclear proteins are also glycosylated. Actually, as we discussed yesterday, the bound ribosome synthesizes protein and then it goes in the lumen of endoplasmic reticulum, isn't it? And in the endoplasmic reticulum, there is certain modification in the endoplasmic reticulum as well as in the cisternae of Golgi complex, and then it is packed into Golgi vesicles and lysosomes. Sometimes when it stays in the lysosome, then it is used for intracellular digestion, and when it is required for secretion, then it is transported outside the cell. But some cytosolic and nuclear proteins are also glycosylated. Actually, nowadays, initially people were thinking only DNA is the genetic material, isn't it? If there is difference in the composition or in the nucleic acid, then character is different. But nowadays people are thinking that only DNA is not responsible for variation, proteins, because ultimately proteins should be expressed. And now people are thinking sometimes this carbohydrate variation also plays an important role. Actually, I will describe all those things in later stage. Glycosylated things should be synthesized on bound ribosome, isn't it? Because when it is inside the, only then it should be. But in certain cases, even in the cytoplasm, there is certain modification. I will describe in the last part of this how it is modified. Rich in information, forming highly specific sites for recognition. Initially, what was the concept? The information is present only in the sequence of nucleotides. But as we know, when there is cell-to-cell interaction or some factors bind with the glycoprotein, carbohydrate is present there and it interacts with the factor and it transmits signal from outside the cell to the inside the cell. It means it has capacity to bear the signal, to transfer the signal, isn't it? Therefore, it is also considered rich in information, forming highly specific sites for recognition. When cell-to-cell interaction, then there is when there is antibody generation due to that recognition only. No? When or autoimmune diseases develop due to self or non-self recognition. If it recognizes self, then there is no autoimmune disease. If it recognizes non-self, then there is autoimmune disease. High affinity binding with carbohydrate binding proteins called lectins. Lectins are those proteins that bind with the carbohydrate with high affinity. And that's why people are thinking these carbohydrates or whatever information, as we know, when there is a particular sequence of nucleotides in the mRNA, then a particular type of protein is synthesized by the ribosome, isn't it? So it means that information is in the form of the code, we say codon, isn't it? Codon for, but that codon is translated by ribosome, isn't it? Like that, lectin binds with the carbohydrate with high affinity, and it reads whatever information is there in the carbohydrate. Therefore, nowadays, actually, just now I was checking on PubMed and I found some reviews people have published even in 2010 and 2011. There are certain information even in carbohydrate, and what is the basis of them. So, as we see, the variation, individual to individual variation is very high. If we consider only at nucleotide sequence level, there are only 4 tetranucleotide, adenine, guanine, cytosine and thymine. If we go at protein level, how many amino acids? 20. And if we go at carbohydrate level, then we will have to consider the monosaccharides. How many types of monosaccharides? 8 and 16. 24. Isn't it? 24 monosaccharides. Different types of monosaccharides. And these monosaccharides in different types of linkage. Isn't it? Different types of linkers. 1,2 linkers, 1,4 linkers, 1,6 linkers, 2,3 linkers. No, different types of linkers, glycosidic bond forms. So you can, on the basis of permutation combination, you can think how many types of combination can form. Therefore, people have hypothesized this lectin acts as a code reader. As in the case of mRNA, code reader is a ribosome, like that. For carbohydrate, code reader is lectin protein. And what is the importance of it? We will study later. Carbohydrate is attached its anomeric carbon through a glycosidic link to the OH of serine or threonine residue, as I mentioned. In most of the cases, kinases are either serine or threonine or tyrosine, or through an N-glycoside link to the amide nitrogen of asparagine aldehyd. And the ratio, if there are 16 linkages, then out of 16, 15 are either serine or threonine, only one is asparagine. Therefore, most of the cases, kinases are serine kinases or threonine kinases. Now, mucin, actually, as you know very well, saliva. Saliva contains mucin. Quite a lot, in our buccal cavity or oral cavity or mouth cavity. There are 4 types of salivary glands, isn't it? So these salivary glands secrete saliva, and most of the part of saliva is made up of mucin. Okay. Mucin secreted or membrane glycoprotein. Actually, many people asked me when we take food, there is secretion of enzyme. There is secretion of enzyme and it digests our food. Proteinous food is also digested by different type of proteases. When we take meat, it is digested, isn't it? When we eat meat or chicken or mutton, it is digested. But why not our digestive tract is digested? Isn't it? If you take the digestive tract of a goat or a bird, you eat it, it is digested. Why not our own digestive tract is digested?
So it is lined with mucus lining.
Mucus lining. That mucinous part is there, that is slimy and slippery. No? And there is also, people always think, when there is infection of Ascaris lumbricoides or Tinea solium or Tinea saginata, it stays in the alimentary canal. It is also not digested because its outer surface of its body is made up of cuticle, and the cuticle is not digested by our enzymes. Due to which, so this mucin plays an important role. It makes, when it mixes, when there's secretion of saliva, it mixes with the food. When we chew it, it becomes a smaller in size, and it mixes, there is enzymatic action, and then it becomes more slippery. It is very easy to swallow, isn't it? So that is made up of the secreted or membrane glycoprotein, large number of O-linked to oligosaccharide chains present in most secretion. It is present in saliva, it is present in gastric juice, it is present in intestinal juice. All places mucin is present. Sequences for the attachment of O-linked chains tend to be rich in glycine, valine and chlorine residues. These are amino acids that are there. Now, glycophorin, first well-characterized membrane glycoprotein, present on the membrane of erythrocyte. The first contains 60% carbohydrate by mass in the form of 16 oligosaccharide chains, and 15 of the oligosaccharide chains are O-linked to serine or threonine, and one is asparagine. That's why for cell signaling, wherever there is need of phosphorylation, there are kinases, and most of the kinases are serine or threonine kinases.
I have a doubt actually in the back. So, do we know, is it genes regulated which are synthesizing mucin? There are two doubts actually. One
Actually, around 1% of total genes plays important role in synthesis of carbohydrates.
1% of the total
1%
genes.
of total genes.
Okay. So the second question is, in the because you are from developmental biology, when exactly this carbohydrate chains or these glycoproteins start forming in the development from the embryo start onwards?
Actually, when there is development of tissue, at that time there is need of extracellular matrix. Initially, when there is blastocyst, at the blastocyst stage you will not get it.
Yes,
there is no requirement of it.
Mm.
In the later stage, when the three-germinal layer is differentiated into different tissues, at that time these genes will express, and then extracellular structure will develop.
My basic aim of this question is that can we artificially
I will come to that point. I will
generate
explain about it. What is the use of carbohydrate can be used for the artificial engineering, or you can say tissue engineering.
Yes.
And genetically engineered cells.
Yes.
No, we actually According to this concept, whatever people are reporting now in last 2-3 days,
Mm.
2-3 years, carbohydrate or extracellular matrix plays an important role. Only then cells express particular type of genes.
Oh.
So if you engineer extracellular matrix, it means carbohydrate. Most of the part is made up of different types of carbohydrates. Of course, proteins are also there, but it also plays an important role. If it is engineered, the gene expression of cells can be changed. Whatever content in each and every cell, there are 46 chromosomes, or in between 30 to 40,000 genes. So all genes will not be expressed at a time.
Yes.
The expression of particular genes depend on the interaction with the extracellular matrix. So if extracellular matrix is engineered in such a way, suppose we have to make a piece of bone, and we are taking mesenchymal stem cell. The mesenchymal stem cell can form fibroblast, it can form adipocyte, it can form osteoblast. If we have to make bone, then it should be differentiated into osteoblast. So if we give that type of a scaffold or particular structure where it binds and it will start to synthesize the extracellular matrix or bone proteins required for the bone and hydroxyapatite crystal, then it will form the bone. If we give different type of scaffold, it will be differentiated into fibroblast or in adipocyte. It means only genes are not sufficient to make a particular tissue. So therefore people are thinking only is information in the gene or is information outside the cell, because expression of genes is depending on interaction with the extracellular matrix.
So mucin is not one gene related.
It is not.
One gene. So I cannot identify the gene and express in some microbial thing, and finally I get a slimy thing like this.
For a particular species, you can, because individual to individual it varies. In certain cases it may be a particular gene. You cannot say okay, it is not, because in our bodies it is secreted by the salivary gland, it is secreted by the gastric gland, it is secreted by the intestinal gland. So place to place its gene will be different. But when you consider a particular microbe, suppose a bacterium, so there may be a specific gene in that case.
No, that specific
If
gene,
you consider all mucins, of course, genes will be different.
if we, mucin is a carbohydrate chain which is linked to a protein.
Yeah.
So that carbohydrate chain is formed afterwards, or how do I ask this question? Can I say that like protein, I can also create mucin as a separate entity? Is it so simple?
No, it is not so simple.
Carbohydrates, it won't be so simple.
It is, you can say, more complex than that of
Correct.
protein. Because both things are involved here. So it is not so simple.
I'll rephrase this. Okay. The question is, in vitro, can I synthesize mucin using our traditional approach of some, say, n number of genes are expressed, and can I put a signal where it will be sent out of the
Artificial particular arrangement of molecules. No? People are trying those things in labs to synthesize artificial synthesis. People are doing, particularly for this genetic engineering thing, they are developing different types of scaffold on which they are loading cells. Accordingly, the cells are differentiated. So people are doing that. To show the similarity with it. It mimics like that structure. Okay. To go to the next. Now, glycolipids. Carbohydrate will be attached with lipids, particularly in our cell membrane. Membrane sphingolipid. Hydrophilic head groups are oligosaccharide. that acts at specific sites for the cognition. As we know, always when we say lipid, amphipathy, head is hydrophilic, tail is hydrophobic, and oligosaccharides are present towards head end, not at the tail end. Brain and neurons are rich in glycolipids, help in nerve conduction and myelin formation. Glycolipid. You know myelin? What is it? For insulation. It is present on the nerve cell, and it is absent at nodes of Ranvier, and it helps in particularly in myelinated neurons. There is saltatory conduction, and it helps in fast conduction of impulse.
What is the disease which doesn't have this myelin sheath? Some disease
Actually,
very famous.
Yeah. Just now it has escaped from my mind. Particularly in children, that is a different case. No, that is a disease, particular disease.
Yeah, it is very famous. Very commonly asked this question.
When there is loss of myelin sheath, then
the
there is a
disease.
disease.
What is the disease?
It has escaped from my mind. Actually, in children.
It is in children actually.
In children, when there is no myelin sheath, initially there is no myelin sheath. Perhaps I describe it. No? There is bed waiting in children when their neurons are not myelinated.
Mhm.
In the later stage, along with the development, the neurons become myelinated. Only then it is able to control the detrusor muscle of the urinary bladder, even in the sleep stage. But when it is non-myelinated, it is unable to control that detrusor muscle movement. Also play a role in signal transduction. You know about signal transduction? Now, gangliosides. Membrane lipids of eukaryotic cells in which the polar head group, you know it, part of the lipid that forms the outer surface of the membrane is a complex oligosaccharide containing a sialic acid. I described it. Sialic acid was negatively charged. Determine blood group. Blood group is dependent on it. Now, aggrecan. Actually, aggrecan is nothing. Simple aggregation of many molecules. Enormous supramolecular assemblies of many more proteins, all bound to a single molecule of hydrogen. That is called aggrecan. Multiple chains of chondroitin sulfate and creatine sulfate are joined to serine residues in the core protein through trisaccharide linkers. Now, these are mainly, you can say, they assemble things at one place. Interacts strongly with collagen in the extracellular matrix of cartilage. Actually, when there is formation of a particular structure, there is need of arrangement of molecules in bundles. So for that, aggrecan is useful, contributing to the development, tensile strength and resiliency of cartilage. Cartilage is elastic, and there should be more, you can say, many molecules together in a bundle. For that, aggrecan is useful. Now, lipopolysaccharides, outer membrane of gram-negative bacteria, targets of the antibodies produced by the vertebrate immune system. What happens when there is infection of a particular bacteria? Then our B cells, or B lymphocytes, when it is exposed to a particular type of antigen, only then it starts to synthesize antibodies. For that, there is a lipopolysaccharide, important determinants of this serotype of bacterial strains. What type of, you can say, antigenic determinant dependent on it. What type of antigen is, accordingly there is a production of antibodies. A portion of lipopolysaccharides of some bacteria is called endotoxin. Toxic shock syndrome is due to infection of gram-negative bacteria. When there is sometimes, it does not harm, only that particular antigen causes shock for the host, nothing else. Now, glycomics. We studied initially glycobiology, now glycomics. systematic characterization of all of the carbohydrate components of a given cell or a tissue. That is glycomics.
How many think future is glycomics? There is lot of future for glycomics. How many are thinking?
If you are not thinking from today, now you will think.
You should think.
Because a lot of things are here.
My God.
Sugar coat. Till now we were studying genetic code, isn't it? Now, sugar coat. Cells use a specific oligosaccharides to encode important information about. When there is a specific type of cell, so on its surface there will be a specific type of oligosaccharide, isn't it? That oligosaccharide has a specific type of information. Accordingly, it will interact with the extracellular matrix, and accordingly there is signal transduction, and finally genes present in the nucleus are expressed. So intracellular targeting of proteins, cell-cell interactions, cell differentiation, tissue development, and extracellular signals. All these are dependent on sugar coat. It is wrong, not 14, 24. 16 plus 8. 24 monosaccharide residues of 4 different kinds. 4 different kinds. Alpha configuration, beta configuration, D form, L form. Understand? The 4 different kinds. Various linkage. 1 2 linkage, 1 3 linkage, 1 4 linkage, 1 6 linkage, 2 3 linkage, and 2 6 linkage. It means total 6 linkage. Now you can think permutation and combination. How many types of carbohydrates can be synthesized in the nature, or is synthesized by the nature? Then how much chance is, how much it will contribute in the variation? Till now we are considering only sequence of nucleotides and sequence of amino acids for the variation. We consider this is the genetic code, and accordingly there is variation. But these oligosaccharides and polysaccharides are also affecting the cellular differentiation, recognition, everything. Alpha and beta configuration. Branched structures are not found in nucleic acid or proteins. This is only simple linkage thing. Alpha, beta configuration, DL configuration, and all these linkages. In protein, amino acid is in the form of a chain only. In the nucleic acid, nucleotides in the form of a linear chain. There is no branching. But in carbohydrates, there is branching also. It means variation due to carbohydrate can be more in comparison to nucleic acid or protein.
With this understanding, of this complexity, can we take these bamboo strips and create the membrane? Is it possible for us to simulate it? Bamboo or something, some wires. Can we really physically see how complex it is? At least to the extent of complexity we can understand.
Yeah.
Can we create a membrane?
Yeah. Actually, bamboo stick, in the piece of bamboo there are many fibers.
No,
Many.
those bottle bottle.
Basket. A small basket.
Basket which is made with a
Yeah.
basket which is made
That is made up of, actually there are many bundles. Yeah.
Can we use that to really simulate a membrane, not the fluid mosaic now, above that whatever is happening, the whole network, because I am not able to feel that complexity that can happen because of branches as well as the linear polysaccharides. Unless we have that information in our mind, we cannot answer any tricky question if somebody asks.
On that, as I think, better than the bamboo thing.
Anything.
If you take the sections, thin slice of bone,
My God.
that is more, it gives more clear cut. I make the structure, then you will.
Did my question make sense, first of all? I always keep wires with me, but the only thing is I'm thinking how to intertwine these wires so that I will see that complex, the phenomena which saccharides are creating, carbohydrates are creating. Even if we go 2% or 3% towards understanding that complexity, we will be able to appreciate how cell is creating this whole thing.
Actually, as you know, osteon is a unit of bone, and in this there is a central blood vessel that is called Haversian canal. And around the Haversian canal there are circumferential lamellae. Like that, there is another osteon. Okay. These lamellae are made up of, as you mentioned, that bamboo stick thing.
Okay.
When we make thatch,
Huh.
in particular tile roof, initially we make thatch of bamboo, isn't it?
Correct.
That is, you can say, a skeleton, and on which we put tiles. So the structure of bone is also like that. Actually, there are longitudinal, you can say, bundles, cylindrical, yeah, bundles of proteins, collagen proteins. Collagen is a fibrous protein, and this is synthesized by cells. As you know, osteoblast. When osteoblast becomes mature, it is called osteocyte, and it is surrounded by its own covering, and there are certain branches. That is called lacuna, plural lacunae. Osteocytes are present in lacunae, and whatever this lamellae is present, it is secretion, or you can say it is synthesized by those osteoblasts. When they become mature, then they are called osteocytes. So then the whole structure is in the form of a bundle. When it and two osteons, this blood vessel is called Haversian canal, and two Haversian canals are joined by Volkmann canal.
Oh, it is exactly like our banana trunk.
Yes.
Huh? Oh.
But in banana trunk there is no
There is no,
horizontal,
horizontal
you can say
action.
transverse
Yes.
connection is not there.
Yes, yes,
But
yes.
in the case of bone, there is a transverse connection that is called Volkmann canal. And this is Haversian canal. And at place to place there is some branching to supply the other. Actually, these will get only these cells, but when there is branching, these lamellae will also get nutrients. Without oxygen or glucose or different types of nutrients are required for the proper maintenance of the bone. For that there are capillaries. When there is a particular, when bone becomes weak, a particular unit is removed. The whole bundle. As you mentioned, in the case of bamboo also there is bundle of cellulosic fibers.
Yes.
So you want to
No.
then?
Actually, in one of the old slides, there is one simulation. Can you go back actually?
So can this be simulated in that also?
Huh?
Once you showed that neck, pinion, joints and all,
Which is it? Wait.
in protein is shown, no? That
You
other
go
paper
back.
was there.
Lot back. This one. See, can we create this with our macroscopic infrastructure, whatever. If we want wires, we can get cloth, whatever it is. Can we see this, and whatever theory we are reading, can we put it?
Or you want to make a model, or you want to mimic the exact condition.
So the answer I don't know how to tell, but I want to see this before me like this. Okay, and now if somebody asks, okay, where is this say pectin, amylopectin or whatever it is on the surface of that fluid mosaic model, and how it is interacting, and which protein, and so take some small protein, and to that where it is getting branched. If I can understand at that level, then it will be permanently in my memory. I don't need to memorize this.
Actually, to know the whole structure. No? Again, the thing is different, tissue to tissue.
Huh?
No?
Let us simplify ourselves. One cell and one cell membrane like this. Forget everything.
Basic thing you can put.
Let us take this itself, and on this we will start adding that if this particular carbohydrate or glycoconjugate or whatever it is there, then it is tissue. Okay?
That can be done.
Huh?
That can be done. That can be done.
Then I will bet that in 1 hour you will know everything about the importance of glycobiology in respect of
That can be done because each and every tissue has its specific characteristics. And on that basis, it is different from other cells, other tissues. So that can be.
This if we can create a model by our own something.
That can be done. But if you take whole, then it will be difficult because in different tissues this arrangement is different.
That's okay.
Extracellular matrix is different, and that glycocalyx, whatever is present on the outer surface of the membrane is.
Once we reach in understanding or simulating this aspect before our eyes, the other complexities can be easily understood. It should not be very difficult.
Yeah,
Because now we are only munching the words, that's all. But at that point of time you have real picture, then it might be very useful.
so you
Don't
can
think
make a
about
model.
computer now.
Good.
Okay? You have to create a model, physical model, then only it will make sense.
So the one which you showed about the proteins was with that to the drag and pinion joints and all. Same thing.
It will be too much complicated if we go at that level. This is now at the level of wires.
So once you brought some wires to the class and you
Yes.
see that you have to
So that we can use.
Now, the reading of sugar coat. Actually, when Rajiv Gandhi was killed, before that there was a message to our IB. IB, do you know IB? Or RAW? You haven't heard IB and RAW? What is IB?
I heard.
Intelligence Bureau, and RAW is Research and Analysis Wing. Both are our intelligence bureaus. Particularly RAW people do not work in India. As you people might have heard about ISI. Have you heard? From Pakistan. Always we tell whatever things occur in India, we say it has been done by ISI.
So ISI is in India now. Like RAW is not in India, ISI is in India.
Yeah, you are right.
Perfect.
Actually, once I was going, that time when it was attacked on Parliament. Yeah, Parliament. So at that time I was going in the train, and one a member of RAW was going with me.
How lucky.
So I asked him, where are you going? He told me, I am going to Rajasthan. Only that, nothing else. At that time, you might have heard, our whole army, almost all regiments were deputed nearby Rajasthan and Gujarat. It was planning of our government if any type of tension occurs, our army will enter in Pakistan directly from that Rajasthan border. So at that time, all raw people will also deputed on that border. So I asked, when initially he was not disclosing, I mentioned ki then where you will go and what will you do? He told, I will not tell you what I will do, but I tell you, I am going there. Even I don't know where I have to go and what I have to do. When I will get down from the train, someone will come to receive me. I don't know who will come. So secrecy is maintained. Actually, I was reading current science. Listen, when I was going to appear at CSIR net. So on the way I was reading current science. Then he understood that I am preparing for net. Then he started to discuss with me what is your aim and what you will do like that. So then he mentioned he will reach there, and then he will have to cross the border, India-Pakistan border. He had raw people cross without passport. You people might have heard. Now many prisoners are in the jail of Pakistan, and India government is not ready to recognize them. Who are they? Actually, most of them are raw people. India cannot say that they are my citizens. They are sometimes they are killed. So I asked, what do you do? Then he mentioned that I have to cross the border. I have to go to Pakistan, and I will stay in the village of Pakistan. People might have seen a film that I forgot that name. There is Abhishek Bachchan and
Refugee.
Refugee. Yeah. People used to cross the border. So he mentioned he will cross the border and he will stay in the village and he will collect informations. And to collect those informations, India government give money to raw people and they give that money to residents or villagers of Pakistan, and then they collect information and they send to India. But if they stay in the capital, suppose Islamabad or in Karachi, so they do not communicate. Some people, sometimes you might have heard, sometimes they elevate, or it is reality, some people from the embassy are kicked out. You cannot stay here. They are doing some secret things. So that is, of course they do. They are deputed only for that.
Yes.
So whatever information they send from Islamabad to Delhi, it is not like that person will be killed. They send. About Rajiv Gandhi, it was told that after some time, particularly in Hindus, after the death, the hair is removed. So in that code it was mentioned before his death that our prince, due to our prince, hair will be removed. Now, at that time who was prince? Rajiv Gandhi. He was the youngest prime minister. At that time he was not the prime minister, but he was considered as prince because he is from the family of Gandhi Nehru. So it was in the coded form, but people sitting here could not decode it, because if you write directly, whatever things are sent at first, that is seen by those people, only then it is sent. They cannot send it directly. It was in the coded form. The message reached, but in the coded form it could not be decoded. Therefore, people did not take extra precaution and he was killed. So, whatever code means it is not in the direct form. Our Vedas or Puranas also in the coded form. Many things are written but people are unable to decode it. They have not so much idea. So whatever information, either in DNA or in RNA, it is decoded by ribosome. Isn't it? When DNA gives message to RNA and RNA reaches to ribosome, then it is decoded in the form of protein. And ultimately what is visible? Character. The protein affects the character. Here also, whatever sequence of monosaccharides and whatever modification is in the monosaccharides, what will it do? That is a code. You cannot say. Then ultimately the decoder is lactin. Lactin will decode it. What that particular type of oligosaccharide will do. Understand? So lactin is found in all organisms that bind carbohydrates with high specificity and with moderate to high affinity. N-acetylglucosamine is recognized by lectin of hepatocytes. As you know, there is glycoprotein, and in that glycoprotein there is N-acetylglucosamine. Generally, you people might have heard, when there is secretion of hormones, such as luteinizing hormone, follicle stimulating hormone, then thyroid stimulating hormone. These are glycoprotein. They come in the circulation, and after some time they disappear, or its level goes down. How its level goes down? What is the reason? So for that there is a specific type of oligosaccharide attached with that hormone, and that is identified by lectin of hepatocyte. Lectin of hepatocyte recognizes. What is that? N-acetylglucosamine. Even that particular hormone, if it is glycoproteinous in nature, and N-acetylglucosamine is present, that will be recognized by the lectin of hepatocyte, and then it is taken inside the hepatocyte and it is degraded. Understand? So that is the process of recycling. You can say sometimes level goes and sometimes go level high. If there is no degradation, level will be high. If there is high degradation, level will be low. So here lectin is playing in, but lectin cannot do its work alone. For that there is need of a specific type of monosaccharide. Here it is N-acetylglucosamine. If it is N-acetylglucosamine, only then it will catch it. It means coded message in the carbohydrate, not in the lectin. As coded message in the mRNA. A particular mRNA sequence is there, therefore particular ribosome binds with it and then it is translated. Isn't it? N-acetylnuraminic acid at the ends of oligosaccharides of many plasma glycoproteins protect from degradation. If N-acetylnuraminic acid is present, then it will not be taken. Understand? But in many diseases, these things become different type of oligosaccharide. Suppose plasma glycoproteins should contain N-acetylnuraminic acid, and when there is disease, it is not N-acetylnuraminic acid, it is N-acetylglucosamine. Then what will happen? Hepatocytes will take those plasma proteins that are required for our bodies, but due to, we can say, misprinting or miscommunication, because the sequence is different, that's why that particular type of glycoprotein will be degraded. Then there is a disease. Now, selectins. Selectins are a family of plasma membrane lectins that mediate cell-to-cell recognition and adhesion in a wide range of cellular processes. particularly when we study about diapedesis. What happens? This is blood capillary, and it is lined with endothelial cells, isn't it? And these are endothelial cells. When there is inflammation, due to inflammation there is secretion of certain cytokines, and these cytokines activate the endothelial cells, and these endothelial cells show some lectin, P-selectin or E-selectin or L-selectin. These are different types of selectins. Particularly this expresses E-selectin. Endothelial cells, E-selectin. What happens when WBC is moving or circulating in the blood vessel? Suppose this is going. I will show the whole animation. In that whole animation, when WBC is moving, when it reaches at a particular point where there is inflammation in the tissue, it stops to move. When it is moving at a particular point, suppose here there is inflammation and this cell is bearing a particular type of hook-like structure, or you can say selectin. Selectin is expressed by the endothelial cells, and when it is moving, the oligosaccharide present on its surface, WBC surface, binds with the selectin. And when it binds with the selectin, then there is cell-to-cell interaction, and then this endothelial cell constricts, and then through this comes out from the blood circulation and reaches to the inflamed tissue, isn't it? So thus, what type of infection is there? Accordingly, this tissue will express particular type of cytokines, isn't it? Even in metastasis also, when there is degradation of the extracellular matrix, there is synthesis of certain chemicals, as I mentioned, MMPs, matrix metalloproteinase, and this communication also. Whatever is expressed here that is responsible for attachment is detached, and then it moves. It goes in the circulation, and then it metastasizes from one place to another place. The P-selectin on the surface of capillary endothelial cells interact with specific oligosaccharide of the glycoprotein of circulating neutrophils, particularly P-selectin. It will interact with the.
So the endothelial cells. It will be on the endothelial cells,
Yes.
and the WBC.
Then it will come out from the blood vessel by the process of diapedesis. Okay? That's all. Some more things. What are, besides these things, what are important? One more day we will discuss. Then it means tomorrow carbohydrate will be over. Okay? And then next session will be for questions. What types of questions can be framed on the whole carbohydrate chapter? Understand? I will put questions here. I will give options, or I will ask answers, anything.